morning. I'm going to discuss some preliminary work that I've done in collaboration with Yongming Sang at Kansas State University for a NIFA AFRI grant. And this is part of a 4-year project in which we intend to study the antiviral regulation and virus-cell interaction in porcine monocytic cells. Activation statuses of monocytic cells, including monocytes, macrophages, and dendritic cells, are critically important for antiviral immunity. Unfortunately, the activation status of porcine monocytic cells, or how cell activation status functionally interacts with antiviral immunity, remains largely unknown. And this is a significant omission because many economically important porcine viruses are monocytotropic, including our focus PRRS. And understanding the relationship of activation status and antiviral states not only extends the activation paradigm of these cells, but also integrates innate immune responses with several aspects of inflammation, tissue repair, and overall antimicrobial activity. And this is important because PRRS infections are syndromes complicated with co-infection of other pathogens. So the integration of activation status and antiviral states would provide a framework for potentiation of overall immune response to PRRS disease. PRRS is ideal for deciphering how monocytic cell activation statuses interact with antiviral immunity. Here we have a list of viruses where they have shown on the macrophage, we see they manipulate and the infection, including HIV, RSV, SARS, and influenza A virus, which may affect macrophage polarization or activation status and lead to immune suppression and or immunopathology. For example, HIV in human monocyte-derived macrophages skews cells towards the M1 or classical-like status, which is correlated with a downregulation of M2 or alternate macrophage activation status markers such as CD163, CD206, CCL18, and IL10, an increase of the M1-associated chemokines or ligands of CCR5, which leads to changes in cell permissiveness and alters the outcome of infection. So our long-term objectives are to integrate activation status with antiviral responses in monocytic cells and to functionally modulate them, or firstly phenotype them and then functionally modulate them for a prototypic cellular adjuvant vaccine that is ideal for potentiating antiviral immunity. To study how PRRS infection alters cell activation, we are characterizing the activation status using polarized monocyte-derived dendritic cells using the framework that's been established for macrophages. Macrophage activation statuses associated with bacterial and parasitic infections are represented by the M1, classical, as I explained. That tends to be stimulated by interferon gamma secreted by Th1 cells. And we have included interferon gamma as one of our mediators. And M1-like pro-inflammatory responses such as would be induced by LPS. The other macrophage activation status is the M2 alternate, and that is stimulated by IL-4, IL-13. We included IL-4, which is secreted by Th2 cells, and then also within M2, there is M2A, B, and C, and M2C-like can be stimulated by IL-10. Interferon alpha boosts antiviral responses through interferon-stimulated genes and pro-inflammatory, but also it stimulates IL-10 and immunosuppressive M2 response. So that's why we've included interferon alpha as a mediator to investigate the activation status using interferon alpha. We have an established RNA-seq procedure for genome-wide identification of signature genes to phenotype the activation status of these porcine monocytic cells. So in this study, we had porcine monocyte-derived or dendritic cells, and they were polarized with the mediators for 30 hours, then mock-infected or infected with PRRSV-infected, a mild strain, and the highly pathogenic PRRS-JX for 5 hours. Each sample represents pooled RNA from 4 replicates. So we did comparisons of the RNA-seq data within each treatment group of activation status, both polarized and unpolarized, using mediator versus PBS control, and between the treatment groups for each mediator. So to remind you, for the mediators, interferon gamma is M1-like. And looking at the macrophage framework, we don't know in dendritic cells. IL-4 is M2-like, LPS, M1-like, and IL-10 is M2C-like. The data was normalized with respect to library size to compare between the paired comparisons. Also, the data was reanalyzed with the data not sized to be more sensitive for comparing the 2 samples. With this, we came up with 33 comparison tables. So, these heat maps show the overall similarity between samples, calculated using both the Euclidean distance and Poisson distance between regularized log transformed data to allow an equal contribution from all genes. When I say regularized log transformed data, we transform the average of the genes across samples to a log2 scale, and that pulls in genes for which the evidence for strong fold changes is weak due to low counts. Once that transformation is applied, the average log expression and regularized log2 fold changes are calculated. The Poisson distance used the original counts, and the technique of multidimensional scaling was used to simplify the clustering. So here we can see that 2H, which is the interferon gamma stimulated high-path PRRS samples, has little similarity to the other samples, whereas the other high-path PRRS stimulated samples are clustering together. We see also the interferon gamma stimulated clustering similarly, and here the interferon alpha for VR2332 and control are clustering together. Also, that similar results are shown. It's just the clusters are moved around. We see, again, interferon alpha for the VR2332 and control are clustered together. There's the high-path PRRS stimulated cells for IL-10, LPS, and interferon alpha. And here's the IL-4 clustered together, the stimulated cells for all the uninfected and VR2332 and high-path PRRS. So this makes it interesting, so we can sort of look at which genes are phenotyping in that they have similar responses or very variable responses across these samples. So that's looking at similarity. Now we're looking at variability using principal component analysis estimates, and we'll emphasize the variations and comparisons. And here, quite simply, we see a lot of variation. Whether a gene effect is significant or called significant depends on not only on its log fold change in expression, but also its within group variability, which is what the DE-seq2 dispersion estimate quantifies. For strongly expressed genes, the dispersion can be understood as a squared coefficient of variation. For weak genes, the Poisson noise is an additional source of noise. So here the black points are the dispersion estimates for each gene as obtained by considering the information from each gene separately. Unless one has many samples, these values fluctuate strongly around their true values. Therefore, we fit the red trend line, which shows the dispersion's dependence on the mean, and then shrink each gene's estimate towards the red line to obtain the final estimates, which are the blue points, that are then used in the hypothesis test. Blue circles above the main cloud of points are genes which have high gene-wise dispersion estimates, which are labelled as dispersion outliers. These estimates are therefore not shrunk towards the fitted trend line. Here we have a heat map of the top 35 most variable genes in the data set. In these plots, we have tried not to do any comparison of differential expression, but rather we're simply looking at each gene across all treatments and asking which 35 genes show the most variability. We then create a heat map using those 35 genes. The logic is that the most variable genes are the most likely to be the genes that will provide the most resolution for clustering the samples. The bar along the top here highlights the location of the 6 mediators, which is reinforced here by the sample names. So these are the mediators, and we're looking at the heat maps of variable genes. In this next slide, we cluster according to viruses, and the bar along the top highlights the location of the 3 virus types, grey being control, red being the VR2332, and blue being the high-path PRRS. Here I have labelled the ensemble names from the current Porcine annotation of these top 35 genes. We see that they're mostly cell components, but I like to highlight the ones that are involved in biological processes. We've got IRG6, which is inflammatory response protein 6, an antiviral, known antiviral for classical swine fever virus. We have CXCL9, which is an antiviral chemokine induced by interferon gamma involved in T-cell trafficking signaling, GCPCR, and the AKT signaling pathway. And then we have tenascin, or TNN, and that's an extracellular matrix, heparin collagen binding. It's also involved in integrin binding. That's part of the PI3K-AKT signaling pathway, which was brought up by Jack Decker yesterday in his talk. Okay, I apologize that I didn't include magnitudes here. These are some genes of interest that we saw most variations in. Between each mediator, again, this is interferon gamma, M1, IL4, M2, LPS, M1, and we see here mainly a pro-inflammatory response. Here IL-10 we see, which is an M2, we see mostly cellular components are the most variable. And interferon alpha, we see biological processes. This is of interest here, some innate immune response genes. These comparisons are comparing control to PBS, or control to PBS, VR232 to PBS, and high-path PRRS to PBS. And I should, as I said, I didn't show the magnitudes, but control, I think the highest fold change was 8, and in our R2, log2, and then increasing fold changes VR232 and high-path PRRS. I would like to further analyze those genes using a gene set enrichment analysis or a leading edge analysis and look at some pathways. Here's the simple comparison pairwise of VR232, each mediator to its control mediator. I'll highlight here we have some inflammatory response genes and interferon-induced transmembrane genes. And in these two sample comparisons, they should be treated as exploratory in nature for screening to pick candidates for further study. Here we compare the VR2332 and the high-path PRRS, and we see here a lot more biological processes, innate immune response genes being upregulated, and antiviral responses. So to conclude, the clustering of samples was consistent with virus strain and then by mediator. Many of the genes showed most variability were related to cellular structure and innate immune response. And the magnitude of differentially expressed gene profiles detected in high-path PRRS infected dendritic cells as compared to VR2332 was consistent with increased pathogenicity of high-path PRRS in vivo. Our future work, we're doing in vivo infection and then looking at the activation status of cells using flow cytometry. And I'm rushing because I'm out of time, but we have several panels set up that we're optimizing because primary cells consist of heterologous populations, and this requires more specific immune reagents for phenotyping these populations. And some antibodies show cross-reactivity to porcine antigens. From these, we'll do some sorting, and we can look at real-time RT-PCR for our genes of interest that we'll have selected from the study. So some preliminary flow data there of showing that we see with the CD172 an increase between 7 and 14 days in the infected pigs. These are PBMCs, and this is from lavage. We looked at CD16 positive CD163, and this is with the MHC2 preliminary flow data. So we're still optimizing that, and we expect we're going to be doing the high-path PRRS study in the same manner, hopefully in the next month or two, and expect a lot stronger results for differentiation of activation status and subtyping of these cells.